Good afternoon, everyone. We're into session 12 on the biosynthesis of antimicrobials. Our first speaker today is Eszter Farkas. She completed her BSc in Translation and Molecular Medicine in 2020. She then began her master's at McGill University under the supervision of Dao Nguyen and co-supervised by Samantha Gruenheid. She then fast-tracked to the PhD in 2022 and is currently a PhD candidate studying the production of antibiotics by Canadian high args bacteria. Besides her graduate studies, she is involved in the McGill AMR Centre Outreach team and science mentoring program.
As mentioned, my name's Eszter. And I'll be talking today about the production of antimicrobials by Canadian high Arctic bacteria. So I don't think I need to impress upon you the importance of the AMR crisis, but one thing I do want to highlight is that currently in the antimicrobial development pipeline, we're really lacking in the discovery of new compounds that could be developed into antibiotics. In terms of what we have currently in the clinic, the majority of the compounds are actually natural products. And many of these were discovered between the 1940s and the 1960s. After this really golden age of discovery, researchers moved on to screening synthetic compounds because they were rediscovering the same compounds over and over from soil bacteria. This was largely unsuccessful, and so now researchers are looking at new avenues, including computational discovery like AI and machine learning, genome mining, new environments, and novel cultivation methods. So as I mentioned, most of our antibiotics are natural products, and these were taken largely from soil bacteria that were found in temperate environments, so essentially the soil from our own backyards. But now researchers are thinking of looking at new environments, especially extreme environments where bacteria are selected for by some extreme pressures that result in them producing some new compounds. Researchers are looking at places like the desert, which have huge differences in temperature between the day and night, have some low nutrients, and lack rainfall, and researchers have already found antibacterial compounds from bacteria and plants in these environments. They also are looking at aquatic environments where there have been antibacterial compounds found from fungi, algae, coral, mollusks, and bacteria, and also some other environments like high salt environments where we have isolated some antibacterial compounds from bacteria. Those are not all of the environments that people are looking at, but there are some examples. And one that we are particularly interested in is the Canadian high Arctic. So this is an area in the north of Canada that has some really extreme conditions, like extreme cold, low nutrient availability, high salinity, and strong solar radiation in the summer months. And so with this, bacteria evolve some unique capabilities. For example, they produce some compounds that protect them against solar radiation. And our collaborator here at McGill, Lyle White, has actually spent a lot of time looking at the microbial communities in this environment. For example, from a paper from his lab, they found that actually there was almost 50% of the relative abundance of bacteria in the Arctic were bacteroidetes, which in comparison to the relative abundance of bacteria in temperate soils, that ranges from about 1.7 to 17%. So there's definitely some differences in the microbial diversity in the north versus here in our own environment. But we're not just looking at what bacteria are there. We're really interested in what are these Arctic bacteria producing. And bacteria from harsh environments, like the Arctic, actually tend to have larger genomes, and this lends itself to more potential biosynthetic capabilities. And you've already heard some talk about biosynthetic gene clusters, which are these groups of genes that are clustered together in order to produce certain compounds. And in terms of bacteria from the Arctic Ocean, 36% of the biosynthetic gene clusters from these bacteria do not actually match any known BGCs in our databases. So there's certainly some potential for new compounds to be produced from these bacteria. And in terms of what's already been found, there have been multiple products from Arctic bacteria that have been isolated that display antibacterial activity, including phenazines, bacteriocins, and silver nanoparticles. So it's really great to look at the Arctic and be excited about the bacteria there, but you actually have to be able to isolate them. And unfortunately, bacterial isolation is a really labor-intensive process. You have to be able to actually figure out the culture conditions under which the bacteria will grow in the lab, and then someone actually has to go through and isolate the bacteria that you find. So generally from some kind of soil sample, you'll get a plate that looks like this top left corner with all kinds of different mixed populations of bacteria. And someone has to go through and pick all of those out. This is not a process that can be automated by robots. And also, one of the really big struggles is that companies are not interested in this work. And so this work largely rests on the shoulders of academics and research institutions. And so to get around this, a lot of researchers around the world are looking at some crowdsourcing initiatives. There's a lot of different initiatives out there, and one of them is the Tiny Earth Initiative, which was started by Joe Handelsman in 2012 at Yale University. It was originally the Small World Initiative, now it's the Tiny Earth Initiative. But this is really an undergraduate-focused crowdsourcing idea where students from all around the world are isolating bacteria from different soil environments. And to date, there's been over 19,000 isolates that have been found worldwide, and there are 22 high-priority isolates and 28 antibiotic structures identified. And so this initiative was incorporated here at McGill in 2015 into an undergraduate Introduction to Microbiology lab course. This was done by Samantha Gruenheid. And since then, each year, about 120 students have gone through to isolate soil bacteria. And they have isolated thousands of colonies each year. It is currently being run by Jasmine Chahal now. And so with all of these different elements, the overall hypothesis of this project is that high Arctic bacteria produce unique compounds with novel antibacterial activity that may lead to the discovery of new antibiotics. And we had two goals for this project. The first was to create an interdisciplinary workflow for antibacterial screening and discovery. And the second was to incorporate the Tiny Earth Initiative into this workflow. So I've already told you about the labor that comes with isolating and culturing bacteria, but really the whole process of antibiotic discovery, especially in an academic research lab, is really labor intensive. Like I said, we don't really have access to all these different kinds of robotics and all these personnel. And so, you just have these grad students who have to go through and screen antibacterial activity and go through a dereplication process to make sure you're not rediscovering the same compounds. And then you have to figure out what kind of antibacterial compounds you're finding. And this is the overall pipeline that we came up with for this project. And I'll go through it throughout the rest of this project. But it starts with sampling in the Canadian High Arctic by our collaborators in the White Lab. They take the soil from all the way up here in Axel Heiberg Island at McGill's Arctic Research Station, and they bring this soil back to McGill and give it to the students of the Tiny Earth at McGill lab course. These students are then the ones who have to go through and isolate them and really go from a mixed bacterial population to a single strain culture in order to have all these different distinct isolates. And this was started in 2017, and since then, every year except for 2020, we've had students doing this isolation. And they generally do it in tryptic soy agar and incubate for 1 week at room temperature. Students are encouraged to isolate bacteria that display interesting morphologies, but really, they are undergrad students. They're not highly skilled researchers, and so they kind of pick whatever interests them. Once students have isolated a number of different Arctic isolates, they then go on to screen them. And this screening is done against escape relatives. The escape pathogens are the leading hospital-acquired pathogens. And since it's a teaching lab, we can't use those pathogens, but they use the safe relatives of these pathogens. And what they do is they grow a lawn of these relatives on an agar plate, and then they patch on their Arctic isolates, incubate them, and look for a zone of inhibition, which you can see down here. Any isolate that is able to inhibit at least one of these relatives is considered to have passed and to have some kind of activity. Any of the isolates that have activity are also 16S sequenced in order to identify what kind of isolate they have. And interestingly, of the ones that have activity, over 50% of them to date have been pseudomonads. So from the 4 years combined, we've had 469 isolates with activity, and that's out of thousands and thousands of colonies that were isolated. And so we then got these isolates, and we put them through a dereplication process. This dereplication process was done with the antibiotic resistance platform, which you heard about on Sunday, which was developed by Jerry Wright's lab. It's a collection of E. coli strains that contain plasmids that confer resistance to different antibiotics. The idea is that if an isolate is able to inhibit all of the strains within this collection that we're testing against, it's potentially producing some kind of novel antibiotic. So one of the things that we did was we had a couple different people working on this dereplication, and we noticed high variability between people, and it was also a very low throughput assay. And so we went on to optimize this assay by developing a replica plater screening assay. It's very simple. We take a rectangular petri dish with agar in it. We grow a lawn of our tester strain. In this case, it's the ARP strain that we're interested in. And we stamp on the Arctic isolates that were grown in a 96-well plate. And then we incubate and look for a zone of inhibition, which you can see down here. So again, we were looking for Arctic isolates that inhibited all of the ARP strains within this collection. And of the 469 isolates, we had 44 that were able to do so, which was, a large atrophy rate, but it was still really exciting to have 44 isolates to work with. We then moved on to screen them against the escape pathogens themselves. Again, we used our replica plater stamping assay, except this time the lawn was whichever escape pathogen we were testing against. And again, we were looking for activity against at least one of these escape pathogens. So here's the collection of all of the 44 isolates and their activity against the various pathogens that we tested. So on the bottom, we have the pathogens, and on the side, we have the zone of inhibition, the number of Arctic isolates that inhibited that pathogen. And out of the 44 strains, we had the majority which inhibited multiple different strains of Staph aureus, both methicillin susceptible and methicillin resistant. And then what was particularly exciting is that we saw a number of isolates that had activity against gram-negatives like Klebsiella pneumoniae and Acinetobacter baumannii. All of the strains are also able to inhibit E. coli delta bambi delta tol C, which is a hyperpermeable and efflux deficient strain, which was also developed by the Wright lab. And this is just a tester strain. So if an isolate isn't able to inhibit this E. coli strain, it's likely not going to inhibit anything else. So from the 44 isolates that we tested against the pathogens, 34 of them inhibited at least one escape pathogen, and we went on to whole genome sequencing. This was done for a number of reasons. One was to actually be able to identify the species and the strain of the isolate. The second was to identify identical isolates within those 34 in case we had duplicates. And the third was to potentially figure out if there were any biosynthetic gene clusters of interest within the genomes of these isolates. So to date, we've analyzed 15 out of the 34 isolates, and 8 of them were considered unique strains, with 7 being eliminated due to being similar to one of the 8 isolates. And of those 8 unique Arctic isolates, we identified 84 biosynthetic gene clusters. Out of those 84, 66, so almost 80%, had less than 40% homology to any known biosynthetic gene cluster. So there's definitely some potential there for new compounds to be produced. And we have 19 isolates that are still in progress. So then we moved on and took those 8 that we've already worked on and wanted to take a look at what kind of compounds they're producing. And for that, we performed organic extractions using ethyl acetate on the cultures of these different bacteria. For the next part of this talk, you're going to see a number of different results from some different assays. We performed some experiments on supernatant from these Arctic isolate cultures, and we screened these against a bioluminescent version of the E. coli delta Bambi delta Tulsi. You'll also see some results from the crude extracts of these Arctic isolates. For this, we had some agar plate well diffusion assays, some minimal inhibitory concentrations, and then also testing against our E. coli strain. And then finally, you'll see some activity or some results from fractions. And again, we tested those in the MIC assay and in the bioluminescent E. coli. So the first step in this process was to actually prioritize which of the extracts from these isolates we wanted to focus on. And for this, what we did is we took the extracts from all of these different 8 isolates and we tested them against the E. coli delta Bambi delta Tulsi. And we did this with an agar well plate diffusion assay. And on the bottom, I just have the different Arctic isolates and their codes. And then on the side, I have the zone of inhibition that these extracts produced against the E. coli. And for the rest of this talk, I'm going to focus on this MNAC strain, this first one in the black bar. And so here I have the results of the activity of this extract against the different pathogens that it had previously shown activity against. And you can see right away that it had activity against multiple strains of Staph aureus, as well as some activity against Enterococcus and Acinetobacter. So this is really exciting, but I was seeing a lot of variability in terms of the extracts that I was getting. And so I went through a rigorous optimization process of how I cultured my bacteria in order to get the most antibacterial production as possible. I optimized for the temperature at which my cultures were grown at, the volume of the culture media, as well as the length of time it was cultured for, and then also the media that it was cultured in. So for most of this pipeline you've seen so far, all these cultures were grown in tryptic soy broth, but I compared this to R2A, which is a minimal media. So here, what I have is the IC50 curves of the supernatant from cultures grown in R2A or TSB against our bioluminescent E. coli. And what you can see right away is that the IC50 from the supernatant grown in R2A was at a much lower concentration than for the one grown in TSB. And when I performed organic extractions on these 2 supernatants, the extracts from these 2 conditions had very different MICs against a number of different pathogens, with the R2A extract being stronger against all of the different pathogens. So now I had my optimized culture conditions and I had a strong crude extract, and I wanted to figure out what actually is within these what actually is in this crude extract that is producing this antibacterial activity. So we moved on to compound investigation with the help of the Eau Claire lab here at McGill in the chemistry department. And we used liquid chromatography to separate out the different compounds based on their polarity. And with this, we did bioactivity guided fractionation. So essentially, we did some chromatography, and then we got our different fractions. We tested all of them for activity, and then the one that had activity, we fractionated further, tested those fractions again, and it was a very reiterative process. I'm not going to show you the entire fractionation process, but I'll just show you a little piece of it. And so here I have the different fractions on the bottom, and then on the side I have the MIC values of these. And you can see right away that fraction 1,7 had the strongest activity with an approximate MIC of about 16 micrograms per milliliter. And when we compared this to the chromatogram that we got from the LCMS when this was being fractionated, that fraction corresponded to these two peaks on the chromatogram. And when we took a closer look at that, those two peaks corresponded to a compound with a mass to charge ratio of 416. And so we went on to isolate this compound with this mass, and then tested it in an MIC assay against our E. coli delta Bambi delta Tol-C. And what we found was that it had activity, which was super exciting, at a concentration of 22.5 micrograms per milliliter. Unfortunately, this is where the story ends for now. We have not yet figured out what this compound is. That's the next step. We're also going to isolate more of this compound in order to be able to test it against various pathogens. But to summarize what I've shown you today, we've successfully set up a pipeline for the screening of Arctic isolates for antimicrobial discovery. We've also incorporated the Tiny Earth initiative at McGill into the discovery pipeline. And not only has this increased the amount of hands that are working on this and isolating all these bacteria, it's also been a really, really great resource in raising awareness for AMR within the student population. All the students get really excited to be helping combat AMR and to be kind of doing their part. And on top of that, we've also successfully identified multiple isolates of interest, and we're in the process of figuring out what our bioactive compound in the MNAC extract might be. And what I really want to emphasize is that this was a huge collaborative effort from the White Lab, who has helped us a lot with the Arctic bacteria, the Eau Claire Lab, who has helped us with all the chemistry, and then of course the Tiny Earth students and all of the crowdsourcing that they've done. There's also an antifungal component to this pipeline, which the Shepherd Lab is part of. And I think this is really the importance of this talk, is that in order to get more compounds into the pipeline, especially with all of this resting largely on the shoulders of academics, the way to move forward is to really work together, to collaborate, to crowdsource, and to try our best to fight to have more compounds. Thank you.
So, Luis Motaro from the University of California, Irvine. So, my question is regarding the main bacteria that you follow. So, is that an actinobacteria?
No, that's a great question. It's a Pseudomonas prasekii. Oh. Yeah.
Because as you found out, the growth conditions might completely change, the chemistry that you get out of. And so it might be a bit of time, nutrients, who knows?
Yeah, that's something we've spent a lot of time thinking about, is how are we culturing these bacteria, how are we isolating them? There's definitely some limits in terms of it being an undergrad teaching lab. They do have to kind of optimize it so that the students can get some work done. And also, in terms of my project, we could optimize for some other bacteria to grow, but if it's going to take them weeks and weeks and weeks to grow, it might not be the best way to have a screening pipeline.
So does the active compound have color?
Yeah, well, the active compound does not, the extract does. But once we fractionated it further, it lost its color.
Eszter, so lovely talk, and really nice to see the Tiny Earth initiative and the crowdsourcing strategy to there's safety in numbers, and I really love this aspect of your project, so congratulations. The question I have for you is, I'm really curious about, and maybe this is a question more for Dr. White, but why are there so many pseudomonads in basically the desert?
That's a great question. I'm not entirely sure. I think, Pseudomonads in general, they're pretty good at growing in a variety of different conditions. I think what we isolated is not representative of the what's out there. I think how we isolated the bacteria from the soil selected more easily for Pseudomonads that are able to grow slowly or to grow in cold versus more warmer climates. In terms of why they're up there in the first place, I don't really know. Yeah.
Okay, we have a question in the back.
Sorry, is it working? Yeah. Hi, I'm AMR from Canadian Food Inspection Agency. I'm just curious, in the future, what method you're going to use in order to know the active compound behind the activity? To know the actual compound that has antibacterial activity. So what's the method you're going to use?
So we're going to use NMR in order to figure out the structure of the compound, and then we're going to compare it to compounds in various databases, probably using mass spectrometry to figure out if it's something that already exists or not.
Okay, thanks.
One final question.
So, Jan Kresp, University of Birmingham, I think it's great that you found so many interesting new bacteria and compounds. But I was wondering, with all your prioritization, you're throwing away a lot of things you should really be following up as well by focusing just on one, but you have so many of interesting compounds to look at.
Yeah, so I guess in the talk I focused on this one extract because that's what we chose to kind of move forward with first, but we are working on a number of other extracts and looking at what they have in them. They're just a little bit further back in the pipeline right now.
Hello, everyone. My name is Zhiguo Zhang from Westlake University in Hangzhou, China. Today, I'm glad to share with you why our recent work about potential impacts of anthropogenic activity and antibacterial biosynthesis on nationwide soil resistance from a metagenomic perspective. I will introduce this work from the following three parts. First, I will introduce briefly the background. Antibiotic resistance is one of the most significant global health threats. Without effective interventions, it's estimated that 10 million people worldwide will die every year from the infections of antibiotic-resistant pathogens by 2050. Well, addressing antibiotic resistance solely from a clinical standpoint is insufficient because humans, animals, plants, and the environment are closely interconnected, and antibiotic-resistant genes will circulate among them. So we should combat antibiotic resistance from the One Health perspective. Agricultural soil is the foundation of our human society and is also a crucial component of the One Health framework. So understanding the health risks and the driving mechanisms of soil resistance is vital. It's important to know that soil ARGs are ancient, and not all ARGs will threaten our humans' health. Recently, Professor Tong Zhang's group at Hong Kong University developed a framework for ranking ARG risks. The ARGs that are accessible to humans, mobilizable, and carried by pathogens are considered as high risk. So now we can by using this framework, we can identify which ARGs are high risk, which ARGs are low risk. But what about the distributions in agricultural soil, specifically in China? It's less studied. And on the other hand, from the microbial ecology perspective, natural antibiotics can serve as microbial weapons, giving their producers a competitive advantage in soil environment. And this antagonistic interaction will may promote the development of antibiotic resistance in soil environment. But the relationship between the biosynthesis of antibacterial compounds and antibiotic resistance genes in microbial community level is less studied. So to fill these research gaps, we launched nationwide sampling campaigns of cropland soil in April 2021. And we also collected forest soil metagenome from NCBI for comparative analysis, since forest soil are less human influenced and they're when compared with cropland soils. After collecting samples, we analyzed the physiochemical properties of cropland soils and conducted metagenomic sequencing. And we then performed with a bioinformatic analysis to quantify the abundance of ARGs and the biosynthetic gene clusters. We use the abundance of antibacterial biosynthetic gene clusters to estimate the biosynthetic potential of antibacterial compounds because the natural antibiotics is difficult to detect and quantify in the soil environment. Our results showed that the total ARG abundance in cropland soil and forest soil showed no significant difference, and the diversity in cropland soil is 1.4 times higher than that in forest soil. In contrast, the abundance of high-risk ARGs in forest soil, in cropland soil, is 25 times higher than that in forest soil, and the diversity is 8 times higher than that in forest soil. And among the 50 cropland sampling sites, we can see that the abundance of high-risk ARGs in 21 sites was more than 5 times higher than that in forest soil. And we can see that the human gut virus, their abundance is highly and significantly correlated with ARG abundance. So these results together indicated that human activities or agricultural activities significantly elevate the risk level of soil resistance. On the other hand, we know that environmental microbes are both producers of natural antibiotics and the hosts of antibiotic resistance genes. So we assume that the biosynthesis of antibacterial compounds may affect the soil resistance regarding their abundance and diversity. So we inspect their relationship. To inspect their relationship, we extracted antibacterial biosynthetic gene clusters from MyBig database, and we quantified their abundance in the soil environment. We can see from the map that the biosynthesis of antibacterial compounds is widespread in the soil environment of China. And we further found that the abundance of antibacterial biosynthetic genes is highly and significantly correlated with ARG abundance, and their diversity is also highly correlated. Further compared with environmental factors and human activities and other biotic factors such as mobile genetic elements and the dominant phylum, we can see that the abundance of antibacterial biosynthetic genes most strongly predict the ARG abundance variation. So this results highlights that the significant impact of biosynthesis of antibacterial compounds on soil resistance. We further explored how the biosynthesis of antibacterial compounds affect the soil resistance. First, the microbial producers of natural antibiotics usually use self-resistance genes to protect themselves from their antibiotics. We truly found some several types of ARGs within the biosynthetic gene clusters, and we quantified their abundance in crop soil and found that they contribute to at least 14% of the total ARG abundance. And on the other hand, the excreted antibiotics from their producers will pose selective pressure on surrounding microbes. We can see from the microbial co-occurs network that the antagonistic interaction greatly intensified as shown in the black eye, with the increasing biosynthetic potential of antibacterial compounds. And under this situation, the antibiotic-resistant bacteria will outcompete the antibiotic-sensitive ones, so leading the increase of ARGs in soil environment. After observed these results, we are further curious that whether natural selective pressure will affect high-risk ARGs in soil environment originating from the manual application. By analyzing public metagenome data set from manual application experiment, we found that the abundance of antibacterial biosynthetic genes is highly correlated with the high-risk ARG abundance in soil environment. So this indicates that natural selective pressure may affect the development of high-risk ARGs in soil environment. But I have to say that these results is solely based on correlation analysis. So the real impact, the real influence of natural selective pressure on high-risk ARGs should be experimentally validated in the future. In conclusions, we first found that the abundance and the diversity of high-risk ARGs in cropland soil were much higher than that in forest soil. Second, we found that biosynthetic potential of antibacterial compound best explained the total ARG abundance when compared with environmental variables and human activities. Third, we found that both microbial producers' self-resistance and antagonistic interactions contributed to the total ARG abundance. And in addition, our EMB lab also using quantitative metaomics and phenotypic metagenomics to address the fate, activity, and host of ARGs in wastewater treatment plants and their receiving rivers. So if you are interested in our work, please visit our website. That's all. Thanks for your attention.
So thank you for that presentation. I'm just very curious, do you know anything about whether or not the areas that were sampled, I know you had information on which ones were manured or not, but do you know if any of them were exposed to various kinds of antibiotics or antimicrobial agents as well? Like, is this just a selection process for low abundance resistance genes and biosynthetic clusters, or is there a direct sort of connection with enriching in a different mechanism?
Actually, we annotated the biosynthetic gene clusters and we got the taxonomy information, and I found that most of them are actinobacteria. And we also found that these antibacterial biosynthetic genes are significantly correlated with 10 of 17 of the main types of ARGs.
There we go. Hi, I'm Clayton Markham from Virginia Tech. For your forest soils and your cropland soils, were they the same soil types and located geographically close to each other? Yeah, and pretty much were you controlling for factors so they were similar as possible other than just growing crops type of thing?
Thank you. The sampling site is not adjacent to each other. It had a wide geographical distribution across them, across China. Yeah, there are different types of soils.
Okay,
Yeah.
But lots of types in each category, I imagine.
Sorry?
Lots of types of soil in each category, I imagine, both agricultural and forest. Yeah. Okay.
Thank you.
Thank you. Lyle White from McGill University. So my question is, so you see lots of antimicrobial resistant genes when you apply manure. So I guess you're basically enriching for those genes because of the organisms that are in the manure?
You mean from the manure?
Sorry?
Sorry, I can't get your question.
Yeah. So you have these soils. You apply manure to fertilize the soils, right?
No.
Yeah.
We just sample the cropland soils, but we have no information about their manual application informations.
Okay.
But you can see in the last slide. In this slide, in the red picture, we in order to validate our results, we collect the public metagenomic dataset from the manual application experiment. And in these experiments, the manual application information is well recorded. We know which field is manual applicated and which is not manual applicated.
Okay. I guess what I'm trying to get at, I'm just trying to understand why you see this correlation between the numbers of the ARGs and then the biosynthetic gene clusters. And I'm just wondering if it's possible that the source of both may be coming from the manure applications that basically reside in the soil after the stuff is applied. Would that be possible?
I think it's possible, but so we just want to investigate whether in the community level the biosynthesis of antibacterial compounds will affect the ARGs. Yeah, but we didn't detect the antibiotic compounds, the chemical compounds. We just estimate their potential using the gene abundance. Yeah, thank you.
Good afternoon. My name is Shihai Liu, a PhD student from Chinese Academy of Sciences. Today I'm going to give a presentation on behalf of Professor Yang on the topic of the control technologies of antibiotic resistance in pharmaceutical industry. This presentation includes 4 parts, and firstly, I will briefly introduce the backgrounds. Wastewater generated during the antibiotic production usually contains high concentrations of antibiotics. For example, the oxytetracycline concentrations is around 1,000 milligram per liter in the oxytetracycline production industry. Such a high concentration of antibiotics can kill the bacteria in activated sludge. What's worse, it can also induce the antibiotic resistance in bacteria. So we want to know what's the concentration levels for antibiotics to adversely affect the biological wastewater treatment performance, and how to control ARG dissemination during antibiotic production wastewater treatment. To answer these questions, we set up bench-scale UASB reactors to investigate how biological process respond to the increasing concentrations of oxytetracycline. And we found that anaerobic digestion was inhibited at an oxytetracycline concentration of 150 milligram per liter. At the same time, we also investigated the occurrence of multi-drug resistance during APW treatment, and we also found bacteria isolated from the treated wastewater and downstream river exhibited high resistance to the tetracycline and many other classes of antibiotics. In order to determine the minimum selective concentration for each antibiotic, we designed a batch test. Specifically, the beakers inoculated with activated sludge was packed with different antibiotics at 6 different levels. The purpose is to determine the concentration levels of antibiotics to cause the resistance. As a result, we found that some antibiotics, such as the tetracycline, erythromycin, and spectromycin, can induce the resistance at a very low level, just about 1 microgram per liter. So far, we have found that the antibiotics in wastewater not only can affect the treatment efficacy, but also can promote the ARG dissemination. To solve this problem, we have to it is vital to remove the antibiotics in the wastewater before biological treatment. Although chemical oxidation is very effective in decomposition of antibiotics, however, it is not suitable for the antibiotic production wastewater pretreatment because of the presence of abundant coexisting organic pollutants. The best way is to selectively remove the antibiotics from APW, the antibiotic production wastewater. And it is known that the functional groups of many antibiotics is easy to hydrolyze. However, the hydrolysis rate is usually very low, too low to be used for wastewater treatment. So we develop wastewater pretreatment technologies based on the acid and base catalytic hydrolysis. Using this technology, we found that 14 out of the 18 antibiotics have lost their antibiotic potency with half-lifetime of several hours. And the feasibility of this technology was validated in a pilot system at a manufacturing site. And we also found the enhanced hydrolysis can significantly remove the COD and the antibiotic oxytetracycline, and also decrease the antibacterial activity. Additionally, we can also find that ARGs in the sludge was significantly reduced by adopting this technology. The enhanced hydrolysis pretreatment technology has now been applied into oxytetracycline manufacturing plants, and the antibiotics could be almost completely removed, and the resistant genes could be reduced by 83% in our on-site study. On the other hand, we also developed anaerobic MBR technologies to replace the UASB1 because of the anaerobic MBR does not rely on the granular sludge, so it can tolerate more higher concentrations of solids in the wastewater. We set up the reactors, and we found that the anaerobic MBR reactors can tolerate an organic loading rate as high as 10 gram COD per liter per day, and with the COD removal rate about 65%. And the protein was substantially removed by 81%. Regarding the ARG abundance, we also found that the enhanced hydrolysis coupled with anaerobic MBR can provide a double guarantee for ARG control. The enhanced hydrolysis concept has been accepted by WHO technical brief, and the technical guideline of China's Ministry of Ecology and Environment for pharmaceutical wastewater treatment. Besides, this technology was also included in WHO guidance on waste and wastewater management in pharmaceutical manufacturing, and which will be published very soon. In our future work, we are planning to develop technologies for treatment of different types of antibiotic production wastewater and waste fermentation biomass, and address the risk of antibiotic resistant development in other developing countries and establish effective approach to contain antibiotic resistance in these countries. Last, there are also 3 posters from our labs, and I would kindly invite you to give valuable comments during the exhibition. Thank you so much.